hear the word, when you heard the word chemotherapy, usually what comes into your mind is cancer, right? So chemotherapy is use of any chemical to treat any disease. So it's not only limited to cancer. Okay, so chemotherapeutic agent, this is any drug used to treat any condition. Okay, so we need first to define the following terms for you to be able to, what do you call this, understand better the following slides. Okay, so antimicrobial agent, this is any drug used to treat infectious diseases. So antimicrobial agents are further subdivided into antibacterial agent, which usually kills, okay, bacteria. Okay, and we also have antifungal for fungi, antiprotozoal for protozoans, and antiviral for viruses. Okay, antibiotics, these are substances produced by microorganism that is effective in killing or inhibiting the growth of other microorganism. Okay, so that is antibiotics. So antibiotics, it is produced by microorganisms. So not all antimicrobial antimicrobial agent, okay, or antimicrobials. Okay, so all antibiotics are antimicrobial agents, but not all antimicrobial agents are antibiotic, because antibiotic is, these are the drugs that produced by certain microorganism. Okay, others are produced chemically. Okay, so we have the following examples of antibiotic, um, from the fungi source, or derived from the fungi. We have the penicillin, okay, cephalosporin, and griseofulvin. Okay, these are all fungi source. Okay, how about for bacterial source, or derived from bacteria? We have the gentamicin, polymycin, streptomycin, neomycin, bacitracin, okay, erythromycin, chloramphenicol, and rifampicin. So these are all bacterial source. Okay, so for the historical notes, or may we have a brief history where this antimicrobial drugs started, or this antibiotic. So ipecac is aided in the treatment of dysentery. So for thousands of years, people have been discovering using herbs and chemicals to treat infectious diseases. Like if you didn't experience this in this time, I actually experienced that before, when we have wounds, like if you don't have creams, that time we use the leaves of guava, okay, and then you have to mash it and then apply it on the wound, and it actually heals the wound. So these are the herbs, or the herbal supplements or alternatives that can be used, okay, to treat wounds and certain diseases. So they use ipecac, aided in the in the treatment of dysentery or diarrheal diseases. So native witch doctors in Central and South America discovered this ipecac, and then also they discovered the quinine extract by cinchona bark in plant, aided in the treatment of malaria, okay, caused by mosquito. Okay, so we have Paul Ehrlich. Okay, he was the first to coin the word, what do you call this, antibiotic. So the birth of modern chemotherapy is because of the work of Paul Ehrlich. Okay, he is credited in his efforts in 1800s. Okay, so while he attempted to stain bacteria without staining the surrounding tissue, he speculated about some magic bullet. So that would destroy bacteria but not the normal body cells. So finally, in 1909, he discovered a compound 606 effective in treating syphilis. Okay? So the compound 606, because it was the 606th compound he tested. The technical name for this is arsphenamine, or the salvarsan, used in the treatment of syphilis. So the technical name is the arsphenamine, and the trade name is the salvastran, or the brand name. Okay, so it became the most widely prescribed drug and the most effective against syphilis until the discovery of penicillin by Alexander Fleming. He also he discovered also the rosanilin used to treat African trypanosomiasis. Okay, rosanilin. Next, we have Alexander Fleming. He's a Scottish bacteriologist who accidentally discovered the first antibiotic. So what is the first antibiotic? That is the penicillin. When you say first antibiotic, it is the first antibiotic produced by microorganism or from microorganism, that is from fungi. So in the representation here, the A is the colonies of the Staphylococcus aureus, and it is accidentally contaminated with the colony of Penicillium notatum, which is a fungi. So the B here is the area of antibiotic activity, where you can see the zone of inhibition inhibiting the growth of the colonies of Staphylococcus aureus. So this is the start of the discovery of penicillium from the Penicillium notatum, from the fungi. Okay, so it has an antibacterial property. Next, we have the Sir Howard Walter Florey and Ernst Boris chain. I think they are the ones who continued the work of Paul Ehrlich. They purified penicillin and demonstrated its effectiveness, okay, in the treatment of various bacterial infections. So during World War II, these two biochemists, they were the first to conduct clinical or initial trials of penicillin, and they are successful in their experiment. So we have Sir Howard Walter Florey and Ernst Boris chain. Okay. Next, we have Gerhard Domak. Okay, he was the first to introduce and discover the proton seal, who is effective against streptococcal infection in mice. So proton seal is a red dye was broken down in the body into sulfonamide, okay, or the sulfa drug, which was the antimicrobial agent. However, it is not an antibiotic because it's not produced by microorganism. So proton seal, I repeat, it's a red dye which was broken down in the body through sulfanilamide. It is not antibiotic because it is not produced by microorganism, but the main component, it's actually basically a chemical. Okay, and then we have Selman, Waxman, and Kolitz. They are the ones isolated streptomycin. Okay, so streptomycin is actually the first anti-tubercular drug. Okay, so it is the first anti-tuberculosis drug. He coined the word antibiotic. So he discovered antibiotics, other antibiotics such as the chloramphenicol, tetracycline, and erythromycin. So they are the ones who discovered the other antibiotics aside from streptomycin. Okay, we have the chloramphenicol, tetracycline, and erythromycin. Okay. So for the ideal qualities of an antimicrobial agents, we have the following. It kill or inhibit the growth of pathogens. So when you say they kill or inhibit growth of pathogens, they need to be effective enough to destroy invaders or the pathogens. Okay? If not, they are not effective. Next, they are selective toxicity. When you say selective toxicity, okay, they should not be toxic, or they should be less irritating to human tissues or human cell. Okay. Another is that they cause no allergic reaction to the host. So that's why when patient first, or they need to first introduce the antibiotic, they need to first test the antibiotic through the skin test. Okay? To make sure that the patient has no allergic reaction in that particular antibiotic or antimicrobial agents. Next, stable when stored in solid or liquid form. Okay? So when they are in solid, they need to not to change in their physical component. Hindi dapat mag-liquefy. Okay? Or in liquid, hindi dapat mag-solidify. So whatever is their original form, they should be stable. Okay? Next, they should be microbicidal rather than biostatic. When you say microbicidal, we learned that microbicidal, these are the drugs that usually destroy or kills all microorganisms. Compared when they are microbistatic, they only inhibit reproduction or the growth of microbes. Okay? Another is that they are not excessive in cost or not expensive. Okay? Another is that they should remain in the body long enough to be effective. Okay? And they kills the pathogen before mutation. Okay? So they kill pathogens before mutation. That's why when you are asked, okay, to take the medicine or the antibiotics for 7 days, you need not to skip any dose of it, or if you need to take it 3 times a day, you need not to take it only once or twice. You need to follow the prescribed dosage or instruction by the physician or the doctor. So these are the ideal qualities of an antimicrobial agents. So it constitutes the greatest number and diversity of antimicrobial agents. Okay. For the mode of action, they usually inhibit cell wall synthesis, damage to cell membrane, okay, inhibition of nucleic acid, inhibition of protein synthesis, and inhibition of enzyme activities, which will be explained in the succeeding slide. So when you say inhibition of cell wall synthesis, they need to be considered to be bactericidal. Okay? They need to kill or destroy all bacteria. Has very little toxic effect to host cell. Example of this are the penicillin, cephalosporin, bacitracin, vancomycin, and antimicrobacterial. So they should be bactericidal. Okay? So one prime example of this, okay, inhibition of cell wall synthesis is the antibacterial agent that inhibits this cell wall is the penicillin. Okay, so recall that in the previous discussion that bacteria contain peptidoglycan. Okay, so peptidoglycan is the major component of the cell wall found in bacteria, right? So penicillin and other antibiotics or antimicrobial agents prevent the synthesis of peptidoglycan, which results to the weakening of the cell wall, eventually making the bacteria undergo lysis or bursting. So it causes destruction causes destruction of the bacteria. So this is the mode of action of this following antibiotics or antimicrobial agents. So they inhibit the cell wall synthesis. Okay, so since human cells or living organisms like humans, we don't have peptidoglycan, we are not affected with this antibiotics because they only target those bacteria which have the peptidoglycan in their cell wall. Okay? So I hope you are following and I hope you are getting my point. Okay? So this is now the representation of inhibiting the cell wall synthesis. Okay. Next, inhibition of enzyme activity. So sulfonamide, so a bacteriostatic and competitive inhibitor, inhibits production of folic acid in those bacteria that require para-amino benzoic acid or yung PABA na sinasabi nila. So, example of this is trimethoprim. Okay? So, in the inhibition of enzyme activity, the sulfonamide, which is bacteriostatic, when you say bacteriostatic, it only inhibits the growth of microbes. And when you say they are also competitive inhibitor, okay, they inhibit the growth of microorganism by competing with the enzyme required to produce the essential metabolite. So when you say essential metabolite, this is any substance required for metabolism. Okay, so this is the work of sulfonamide. Okay, so in the representation here, since the sulfonamide, how this sulfonamide works in the inhibition of enzyme activity is that bacteria requires folic acid in order to carry out metabolic processes. In order for them to produce folic acid, they need the PABA. Yung PABA is needed by the bacteria to produce the folic acid. Since sulfa drugs are similar in shape, okay, to that of PABA, okay, para-amino benzoic acid, okay, the bacteria now are being confused, okay, and use sulfa to synthesize folic acid. Since in reality, however, the sulfa cannot be used to produce folic acid, and without folic acid, the bacteria will die. Okay? Kumbaga parang they only alter the shape of the PABA. Parang naloko lang yung bacteria with the shape of the ABA for the PABA, which is like the shape of the sulfa. When you take this antibiotic, in reality, the sulfa drugs, okay, actually don't produce folic acid in humans. The folic acid, we get it from the food that we eat, from the nutrients that we get from the food. Okay, so that's why it only destroys the bacteria, not the human cells or the human tissue. So that's how it works, the sulfonamides or the sulfa drug. Okay. Next is damage to cell membrane. So one of the mode of action of the antibacterial agents is bactericidal. So they kills most of the bacteria. It brings about changes in the permeability of plasma membrane. Example of this antibacterial agents is polymyxin. Okay? They usually damage the cell membrane. So they result in the loss of important metabolites from the cell. Okay? So in the inhibition of nucleic acid, they interfere with the processes of DNA replication. It has actually limited of usefulness because it may, one way or another, okay, may had side effects on the DNA of human or the living organism. Example of this are the rifampicin, quinolone, or the combination of nalidixic acid and ciprofloxacin. Okay, so they are of limited use because they might interfere with the mammalian DNA and the RNA synthesis. Next is inhibition of protein synthesis. So they interferes with the production of ribosomes. So why do these drugs kill microorganism and not the host cell? Still remember that the eukaryotes have different ribosomes. For eukaryotes, they usually have 80 S ribosomes compared with 70. So these following antibacterial agents like chloramphenicol, erythromycin, streptomycin, tetracycline only target 70 ribosomes, 70 S ribosomes. Then don't target the 80 S ribosomes, which is present in humans. So we are not affected with these antibacterial agents. Only those bacteria who is present, who invades our system. Okay, that is inhibition of protein synthesis. For the spectrum of activity, this is the range of different microbial types they affect. So we already heard the term narrow spectrum antibiotics, which only affects either, when you say narrow spectrum antibiotics or antibacterial agents, they usually only destroys or affects either gram-negative or gram-positive. Example of this is vancomycin, colistin, and the nalidixic acid. Okay? When you say broad spectrum antibiotics, they usually affects both gram-negative and gram-positive, and most of our normal microbiota and normal microflora are also affected with the broad spectrum antibiotic. Okay, so it is usually used only when the pathogen is not yet identified, but the therapy or the medication needs to be started. Kumbaga, just to make sure that you need to start now para kumbaga to start destroying and killing the pathogen within, okay, they usually use the broad spectrum to start the therapy or start the medication. That's the difference within with narrow spectrum and broad spectrum antibacterial agents. How about for the multi-drug therapy? This is the use of 2 or more drugs to kill all pathogens. Uses the principle of synergism. You still remember the principle of synergism? This is the combined effect of both drugs produces a greater effect or a greater antibacterial activity compared to that individual action of each drug. So that is the principle of synergism, the use of both or combination of drugs. Example of this is the DOTS or the direct observed treatment short course for TB. Okay, so we have the following combination of rifampicin, isoniazid, pyrazinamide, ethambutol, and streptomycin. So we have the abbreviation RIPES for the treatment for TB. Okay? Are you still following, class? Konti na lang ito. Okay? Are you still following? Okay? We have the abbreviation for the treatment for TB. We have the RIPES. Rifampicin, so it needs to have the 4 or the 5 drugs to treat TB. Kung rifampicin and isoniazid lang, you will not cure TB. It needs to have the principle of synergistic drugs, okay? The 5 combination of these drugs, okay? For the general consideration for the antibiotics, okay, we have the following. Monitor for super infections, especially for those long therapy for those taking broad-spectrum antibacterial or antibiotics, they might be prone to superinfection. Okay? Next, evaluate renal or liver function. Okay? When you say evaluate renal and liver function, when taking drugs or medication, they usually need to 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 observe if you have damages in your renal. We call it the nephrotoxic. Nephrotoxic. Okay? When you taken drugs or antibiotics or antibacterial, may damage your kidney. So they are nephrotoxic or nephrotoxicity. Okay? For the liver, we call it hepatotoxic. When you take drugs or medication that may damage your liver, we call them hepatotoxicity or hepatotoxic. Okay? Another general consideration for antibiotics or antibacterial or antimicrobial agents, do not store in medicine cabinet because it might be, okay, it can reach by, can easily reach by children. Next, inform provider prior to taking other medications. So if you have maintenance dose before they will give you another medicine for your infection, they need to know your maintenance medication, that it might interfere with your or overlapped with your present condition. Okay. Next, cultures prior to initial dose. When you say cultures, they need to know the effectiveness or the effectiveness of that antibiotic. If the antimicrobial or antibiotic will work, they need to know, they need to perform cultures. Okay. Next, ask about allergy. Okay. If you have antibiotics, like what I've said, they need to have skin test first. Okay. And then take full course of the drug. You need to finish the 7th-day medication. Hindi yung 3 days pa lang, oh, okay na ako, wala na akong lagnat, wala na akong sakit, I'm fine, I'm okay, I'll stop the medication. That should not be. You need to take full course of the drug. You need to finish the prescribed dose and medication. If it's for 5 days, 7 days, you need to finish everything. Okay? And then evaluate cultures. Okay? So WBC, the temperature, this is to determine the effectiveness of the drug. Okay? These are the general consideration for antibiotics. For the antifungal drugs, the action is usually they bind with cell membrane sterol. Okay? Interfere with sterol synthesis and black mitosis or nucleic acid synthesis. Okay? So in these antifungal agents, they usually causes cell membrane to be excessively permeable. Example of this is nystatin and amphotericin B. So remember that sterol, they are waxy insoluble substances. So in fungal membranes, they have the primary sterol. Okay, in fungal membrane, in their cell membrane, they usually have ergosterol. For humans, okay, for mammals, they have cholesterol. So the antifungal agent binds to sterol of fungus or the fungal or the mammary in animal, okay, to destroy the integrity of the cell membrane. So they bind in the sterol or fungus or in animals. Okay. Another antifungal agents is clotrimazole or miconazole. So usually we have creams for this, yung antifungal. If you have mga ganun, di ba? They usually use antifungal cream, okay? Like yung mga may azole, ketonazole, okay? These are used as topical ointments for the treatment of cutaneous mycosis like athlete's foot, okay? or the An or Jack Inch. Okay? Or the brand name of this antifungal are yung mga Canesten or the Trocid. If you experience applying those in your wounds. Okay? And then they block mitosis or nucleic acid synthesis. So they interferes with the biosynthesis of RNA. Example is the griseofulvin and 5-flucytoxin. Okay, so they usually disrupts protein synthesis. Okay, for antiprotozoal agents, they interfere with DNA and RNA synthesis. Okay, interfering with of course, with the protozoal metabolism. Okay, so example of this is metronidazole, or the brand name of the metronidazole is the Flagyl. Okay, so if you have yung parang diarrhea caused by amoeba, okay, if you have amoebiasis, you can take Flagyl or metronidazole. Okay, for antiviral agents, the mode of action, they usually inhibit viral replication within the cell. So we have the following examples. The first antiviral effective against HIV is the zidovudin or the AZT, or we usually term it the cocktail drug. Okay, and now in this time of pandemic, we have the first drug used to treat severe COVID is the dexamethasone. And the second drug, which is baka naririnig nyo ito in the Facebook, that some people needs to buy tocilizumab. Ang hirap i-pronounce. Okay, this is the second drug used to treat COVID-19 patient or COVID-19 diseases. So these are the recent antiviral agents that are produced. Okay, ang mahal daw nito 80,000. Yung tocilizumab used to treat severe COVID, yung mga it has an oxygen, they already have lower oxygen content, mga ganun na yung oxygen na severe COVID, they need this tocilizumab. Actually 80,000, because I knew this because one of my colleagues, yung father-in-law niya, nagka-severe COVID, nag-80,000. Though they cannot buy this in the drugstore because it is not fully established. Okay? Kaya maraming nahuhuli yung mga illegal na nagbebenta nito. And because they usually jump the price. Imagine 80,000 for the severe COVID-19 treatment. Okay, for the drug assistance or drug resistance, okay, so we have two type of drug resistance. We have intrinsic resistance and acquired resistance. So when you say intrinsic resistance, okay, this is naturally resistant to an antimicrobial agent. Okay, so for the acquired resistance, when you say intrinsic resistance, it's within, it's natural. Like for example, for Mycoplasmas species, remember, Mycoplasmas, they don't have cell wall. They are cell wall deficient. So this following microorganism are already had intrinsics or natural resistant to antimicrobial agent, because other antimicrobial agent inhibit the cell wall synthesis, di ba? Cell membrane, cell wall synthesis. And since they don't have cell wall, they are not affected. So this is an example of intrinsic resistance. For the acquired, we have two types. We have the mutation and acquisition. And for mutation, we have a change in drug binding site and a change in cell membrane permeability. For conjugation, we have conjugation, transduction, and transformation, which will be explained in the succeeding slide. Okay. For the change in drug binding sites, okay. So remember, okay, the drug needs to be in the correct receptor in the binding site. Okay. So if they don't have, okay, this binding site, okay, they will not be effective on the microorganism and they can be resistant to it. Okay. The change, this is the representation in the change in cell permeability. Okay. Any change in cell permeability, the drugs will not actually act on the microorganism. It can be resistant, can be gain resistant to that particular antibacterial agent. Okay? So if they lack the specific target size for that drug, the drug will not be that effective. Okay? For the acquired resistance, this is now the acquisition of genes. Okay, first is the conjugation or the bacterial sex. This is the primary way of acquiring new genes, the process of conjugation. Transformation, this is the DNA capture from solution or the uptake of naked or free DNA from the environment. Transduction, this is the case of piggy bank DNA. They term it the piggy bank DNA because they carry bacterial DNA from one bacteria to another, the passing of DNA material. Okay. Next, we have the B or the beta-lactamases. The beta or the B-lactamases, these are enzymes that destroys B-lactam ring. If the certain, okay, antibiotics, okay, have the B-lactamase inhibitor, okay, they may be effective, but they may be resistance, okay, because there are what do you call this? There are microorganisms that have B-lactam ring. Okay, so example of this is unacin, the combination of ampicillin plus the sulbactam. Okay, and then we have the augmentin, which is the combination of amoxicillin and clavulinate. Ang mahal ng augmentin, I tell you. So I repeat, we have the B-lactamases, which results to resistance. These are the enzymes that destroy B-lactam ring or the beta-lactam ring. Okay. So there are 4 mechanisms, okay, in which the certain antibiotic may gain resistance, or certain bacteria or microorganism may gain resistance to antibiotics. It has 4 main mechanism, okay, of microbial resistance. First is the blocking entry. Okay. Second is inactivating enzymes. Third is the alteration of target molecule. And fourth is the efflux of antibiotics. Okay. Efflux of antibiotic, when you say efflux, they pump out the drugs. That is the 4 main mechanism of microbial, I should say, microbial resistance. For the drug resistance, we have the term superbugs. Okay. When you say superbugs, these are the microorganisms that are resistant to certain antibacterial or antimicrobial drugs. Okay, so because of the increasing cases of drug resistance, they thought of this. Okay, superbugs drugs. We have the methicillin-resistant Staphylococcus aureus or the MRSA, the methicillin-resistant Staphylococcus epidermidis or the MRSA, and then we have the vancomycin intermediate Staphylococcus or the VISA, and then we have the vancomycin-resistant Staphylococcus aureus or the VRSA. Okay? So, vancomycin-resistant Stereococcus species of VRA, these are the resistant to most anti-enterococcal drugs. Common cause of nosocomial infection or the nosocomial urinary tract infection. The multi-drug resistant Mycobacterium tuberculosis, patient with this strain may have a lung or section of the lung removed. So if the drugs are now no longer effective, they may remove, okay, a part of the lung or section of the lung removed as an alternative treatment. Okay. For the B-lactamase producing strains of Streptococcus pneumoniae and Haemophilus influenzae. Okay? So for the empirical therapy, so this is the therapy initiated by clinicians before laboratory results are available. So when you say empirical therapy, this is the educated guess of the doctor or the physician. Yung sinasabi niya without even confirming it with the laboratory, this is the educated guess or intelligent guess of the physician. Through experience, of course, they may address the diagnosis of the disease. Okay, that is the empirical therapy. What are the undesirable effects of antimicrobial agents? We have, of course, resistance. Okay. These are the undesirable effects of antimicrobial agents. Resistance. And we learned the different factors to how the microorganism resists to certain antibiotics or antimicrobial agents. Next is the allergy development. You can have allergy to some antimicrobial agents or antibiotics. And then toxicity. So remember that these antimicrobials are still drugs or chemicals. The moment you ingest it, the moment you take it, of course, there will be some minor effect or major effect that may destroy our normal microbiota and normal microflora. Okay? Another undesirable effects of antimicrobial is that the superinfection. Okay? Superinfection, if you will not be able to follow the prescribed dosage, okay, they will multiply and you will be able to have not only one infection but multiple infections because of this superinfection. Okay, so these are the undesirable effects of antimicrobial agents. Okay, so I think this is the last. My God, it's already 12:24 class. Okay, so any question regarding the physical agents, chemical agents, and antimicrobial agents to inhibit the growth of microbes? Yes? Yes, anak, Miss Bascunya.
Ma'am.
Can you louder your voice?
Can you hear me, ma'am?
Yes, yes.
My question for ma'am is if an antimicrobial agent is... Ma'am, is my audio clear po?
Yes, yes.
Okay. If an antimicrobial agent is microbistatic, ma'am, can it affect microorganism that is already present or just inhibit the growth of microorganisms, ma'am?
Like what I've said in my previous discussion this morning, if it's microbistatic, it only inhibits growth. It actually not kills or destroys microorganisms. So that's why a lot of antibiotics or a lot of antimicrobial agents are now microbicidal or bactericidal. Okay? Because it needs to destroy or kills microorganism, not only inhibits. Okay? So this depends on what is your infection. If it's a minor, they might give you a microbiostatic drug. Okay? Just to inhibit the uh, the infection, not to continue to a worsen disease or infection. Okay? Did I answer your question, anak?
Yes, ma'am. Thank you
Okay.
po.
Who else? Who has another queries or clarification question before we end this meeting? Because it's already 12:26. I'm so sorry for this 3.5 discussion. But of course, I thank you because we finished the discussion and up to the last topic for the prelim. Any question before we end the meeting? Of course, I will be uploading to your respective teams the PowerPoint presentation, and also I will be downloading the recordings, and I might I might upload it to section F. Tingnan ko kung pwede. Hindi ko pa natatry eh. But I will try it if it can be downloaded and share it in your specific or respective teams. Any questions regarding the discussions? Meron pa? May tanong? So, for my section 1F, okay, this afternoon, I will just meet you at 1:30 if it's okay with you so that medyo makapag-relax kayo. And kasi medyo matagal yung discussion natin. So, I will ask Sir Christian to meet you for my section F to just meet you at 1:30. Okay? For you to have your our lunch first for 1 hour and to have a break for 1 hour para makapag-regain tayo ng ating energy for our next laboratory class. Okay? So if there are no question, thank you. I would like, yes, anak, Miss Bascunye.
Ma'am, I have another question po.
Sige, sige.
May I ask po, ma'am, how many items is the exam and the pointers po?
For your prelim exam lecture, it is actually 100 items. So it's 100 points. So it covers everything that's been discussed from the start until this discussion. Lahat naman nasa PowerPoint. So whatever it's existing in your teams, lahat ng PowerPoints, it's all included in the prelim lecture. For your laboratory prelim exam, this covers all the prelim topics, lab activities in the lab, including the video presentation, video performance. Okay? For the lab, I think also 100 items. Okay? So, any more? Sige, thank you, anak. Wala na? So, can we have now the prayer? Before we end this, and we have the prayer, I thank you, my dear students, for attending my lecture meeting this morning for us to complete everything. Okay, I thank you for bearing this 3.5 hour of meeting together. Okay, wala na akong boses. I still have 1:30 this afternoon. Okay, so thank you for this for joining me here. Okay, Miss Bakay, are you ready, anak, for the closing prayer?